Hello, everybody. Thank you so much for coming to this month's IWGSC webinar series. In just a few minutes, we're going to be joined by Tanushree Halder, and she'll be giving a presentation today. Before we do that, though, I'd like to first give a little bit of an introduction to the IWGSC. So that stands for the International Wheat Genome Sequencing Consortium, and that is an organization designed to organize the wheat community to do science. And right now, members come from 80 countries and 971 institutions or companies, and we have about 3,500 members and 6 sponsors. And these are our sponsors. We want to thank them very much because they make this webinar possible. So they include Arbor Biosciences, CIMMYT, Flore de Mom, and INRAE, RAG, RAGT, and Kansas Wheat. So thank you, sponsors. And just to get a little bit more into the IWGSC, you may know that IWGSC was responsible for the first release of a complete hexaploid wheat genome sequence in Chinese Spring. We are currently working on a new version of that, but also part of the vision of IWGSC is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And part of that is a new project that was launched just about a year ago, and we have just about a year left on this project, but the idea is to explore the diversity, the natural diversity of bred wheat. And so we are conducting a sequence and assembly of 12 landrace accessions, which represent the founder diversity or phylogeographic diversity. And as well, we are updating Chinese Spring. You can expect gene annotations for all of these and also presentation of data in a graph structure such as the practical haplotype graph. So keep your eyes out for that, and we'll continue to make progress. In about a month, our next webinar is on May 15th, and that will be given by Simon Griffiths of John Innes Center, and he will be the title of his talk is Breeding: Why We Need to Start All Over Again. So please register for that and tell your friends to join as well. And very quickly, I just want to describe the interface that you're using right now for the webinar. If you have questions, you can submit questions in writing using the Q&A icon, and we will collect your questions and we will go through them at the end of the talk. Now, there's also a chat window, and that will be used in order to share information with attendees, such as handouts and things like that. And once again, you can register to our mailing list for information about future webinars. Okay, now the main attraction for today. Today we are joined by Tanushree Halder. She's an associate professor at Sher-e-Bangla Agricultural University in Bangladesh, and also has an appointment at the University of Western Australia in Australia. And the title of her talk today is Proteomic Insights into Protein Biomarkers for Root Trait Improvement in Bred Wheat. And so thank you again for your participation, and we'll turn it over to Tanushree.
Thank you, Josh. Can you see it and hear me?
Yes.
Okay, thank you, Josh, for a nice introduction. And I want to convey my cordial thanks to IWGSC for inviting me for presenting my research work. So it is a part of my PhD research, which was conducted at the University of Western Australia during my PhD. During my PhD, I identified util genes and proteins which are associated with the root traits in hexaploid wheat. And today I would like to present part of it, which is unraveling protein biomarkers for improving root traits in red wheat using proteomics. Firstly, I would like to acknowledge my supervisory team, Professor Kadambar Siddiq, Professor Bujunian, Dr. Inglam Chen, Hui Lui, and also Dr. L.K. Stoer, who helped me to conduct this experiment. And I also like to convey my thanks to my lab mates and friends. As this is a wider audience who are aware about the importance of wheat, I will not go for that. I will just highlight a little bit of the production and the challenges for the wheats and what can be the solution. So currently, wheat production in the world is 786.1 million tons, and the demand is increasing. However, in 2024 to 25, it has been forecasted that the utilization of wheat will be increased 1.6 million tons, but the supply will be decreased by 4.6 million tons. And this reduction of this, or like if we want to meet this demand, and also like if we want to go to meet the demand which has been predicted by summit to make it doubled by 2000 to 2050, we need to improve the yield of wheat. However, wheat yield is actually challenged by different abiotic stresses which has been increased due to the climate change, and among them drought, salinity, waterlogging, and heat stresses are primarily affecting the yield and production of wheat. So, what can we do? So, to increase the productivity of yield and to improve the climate resilience, wheat root system architecture play a crucial role because it supplies water and nutrients to the plant, which is essential for photosynthesis. And at any climatic condition, it is important for grain yield. So, by improving the wheat root system architecture, we can contribute to the um, to meet the demand of wheat in the world in near future. However, studying wheat is challenging. The phenotyping is challenging, and for its precise improvement, we need to explore the molecular mechanism that is also challenging. As roots grow under the soil, so to get the reliable data from the soil, we need to uh, go for destructive method, and we lose it, and therefore we cannot get the reliable data. Additionally, soil heterogeneity also affect the root growth and development. And for getting precise molecular data, we need good phenotyping data also. Additionally, for exploring the molecular mechanism, we need to think about the genetic constitution of the root. Root is a polygenic trait, so which is a quantitative, therefore, it is a quantitative nature. And wheat, it has a large genome, it is 17 gigabyte, and it is very complex, having a repetitive DNA. So, how can we improve root system architecture? There are a couple of approaches. Conventional breeding, which is less precise and time consuming. We can go for the other omics techniques, for example, genomics, but we cannot get the uh, complete protein information from the genomics. Transcriptomics, it has been found that 65 to 75% gene which has been identified in transcription level are not going for translation. So also some of the genetic information is missing, while proteomics is an excellent tool to identify or like to unravel the molecular mechanism of root system architecture in wheat, because we can get the precise information about the genes. It can identify the localization of the proteins. After the translation, I can find the information about post-translational modification and other molecular pathways. Therefore, researchers are focusing more into the proteomics technique to get more information about the quantitative traits, including wheat. So if we look at the journey of the proteomics, uh, here, like in 2003, the wheat proteomics has started. However, in root proteomics started in 2005, and it took long time to get an comprehensive root proteome data in 2014. And further, it has been developing. So what's the issue that the proteomic journey is slower with root genetics or like with root proteomics? Because root is a complex tissue, and it has lack of protein extraction methodology, and also like the proteomic approaches itself have some drawbacks. For example, among all the popular proteomic approaches to DE, label based and label free proteomics are have been using in root proteomics. Among all these approaches, label-free proteomics got more attention because it can identify the complete proteins in a precise manner. It is reliable, and also like it's cost-effective. So the journey is now continuing, and thinking about the importance of the identification of the uh, proteins which are associated with different root traits. I have done this project. So we published one review article which discussed about the journey of with proteomics in abiotic stress tolerance as well as in root system architecture. And for this particular project, my objectives were to identify differentially abundant proteins which control total root length and root dry mass in near-isogenic lines of wheat using a label-free quantitative proteomic approach, because among all the root traits, total root length and root dry mass are more important because it contributes significantly to improve the grain yield as well as climate resilience in wheat, and further to identify their molecular pathways, and then to identify any candidate protein biomarkers for the targeted root traits by comparing the mRNA and protein expression. So for materials, I used two pairs of near-isogenic lines. The near-isogenic lines means the isolines which has like identical genetic background but one contrasting genomic region. So for near-isogenic line 6, we used C306 and the were dry as a parent, targeting one QTL for drought tolerant. And for near-isogenic line paired 9, we used W156 and cascades as parents and targeting QTL or like genomic region, which is heat tolerant. And both of the NILs were developed following heterogeneous inbred family and coupled with first generation cycling method. To get reliable root data, I used semi-hydroponic system, which give the homogeneous environment to grow the plants, and also it is economic. It can accommodate a lot of plants in a small space. So the plants were grown here for 20 days to take the tissues. To get the root total root length data, so I harvested them and separated them from the shoot, and then further scanned it and analyzed it using WinRISO software, and the root dry mass data was found after oven dry. For the proteomics method, I used total root tissue, and then protein was extracted using chloroform and methanol precipitation method. And following that, protein quantitation, protein digestion, and level-free quantification, I identified the differentially abundant proteins between the isolines. And further, I have analyzed the pathway analysis using like gene ontology and cake pathways. Further, I investigated the mRNA expression of the proteins using qRT-PCR, and the differentially abundant proteins which showed similar expression in transcription and translation were considered as candidate protein biomarkers for root traits. So we found contrasting root traits behavior between the isolines. Here is nil pair 6A and 6B, 9A and 9B. So in case of total root length, nil 6A had 66.82% higher total root length than the 6B, while nil 9A had 56.86% higher root length than the 9B. For the dry mass, 6A had 73% higher root dry mass than the 6B. And in case of nil 9, 9A has 57.17% higher root dry mass than the 9B. So after the phenotyping data, I went for the proteomics and I found that for nil pair 6, 99 high abundant proteins and 14 low abundant proteins were found. And for nil pair 9, 9 high abundant and 21 low abundant proteins were found for total root length and root dry mass in wheat. Here is the PCA. So we can see that from the principal component analysis, the 6A and 6B isolines proteins were placed in different places. So we can understand that, okay, they have like the protein abundance has the differences. And then digging into the different molecular processes that are associated with the differentially abundant proteins, from the near-isogenic line pair 6, I found 30 biological processes which are significantly different from each other. So you can see that here are the most significantly different one, and the p-values are increasing over here. And for the molecular function, I found 22 significantly different molecular functions for the root traits. And then for the cellular components, there are 15 different cellular components were found for the DAPs, which are associated for the nil pair 6. Moreover, which are most significantly enriched pathways for the DAPs in nil pair 6 were, for the biological process, it was found that cell redox homeostasis, calcium ion transport, protein localization, protein transport, and other metabolic processes of the protein contributes more for the DAPs. For the molecular function, different structural molecular activity, glucose 6-phosphate hydrogenase activity, oxidoreductase activity, binding molecules such as protein, nucleotide, and manganese ions contributed more. And the DAPs are associated with the membrane coat, calcium coat, and microtubule, as well as cytoplasm. So we can see that different molecular mechanisms are associated with these differentially abundant proteins for the nil pair 6. While in nil pair 9, we found 16 biological processes which are significantly enriched. The color intensity actually means that the higher color intensity means highly significantly enriched, and the lower one means the significant it means the significant became lower. So the red one means highly significantly different, and the yellow one means significantly different, but the intensity is lower. And further, 6 molecular functions were significantly different in nil pair 9, and 24 significantly different and highly enriched cellular components are found in nil pair 9. Among all the molecular pathways in nil pair 9, metabolic processes of cell, protein, macromolecules, and other compounds, as well as cellular process and biosynthesis, contributed more to the DAPs. And for the molecular functions, unfolded protein binding, structural molecular formation, molecule binding activities were significantly enriched for the nil pair 9. And for the cellular components, ribosomal subunits, cytoplasm, endoplasmic reticulum, and other membranes are significantly enriched. Both of the NIL pairs have some common pathways. For example, for the biological processes, they share like cellular nitrogen compound metabolic process and small molecule metabolic process. And for the molecular functions, they share these structure molecule activity, protein binding, coenzyme binding, cofactor binding, nucleotide binding, and purine nucleotide binding. Means these shared molecular processes can contribute significantly to increase the total root length and root dry mass. Further, to investigate the cake pathways, we found that both of the NIL pairs actually significantly enriched or associated with genetic information processing and followed by carbohydrate metabolism and cellular process. For NIL pair 6, it was found that aspartate and glutamate metabolism, pentose phosphate metabolism, glycolysis, ribosome pathway are more associated with the DAPs of NIL pair 6, while in NIL pair 9, ribosome, protein export, riboflavin metabolism, riboflavin pathway, TCA cycle, glycolysis are more associated with NIL pair 9. Means they also share some of the pathways such as glycolysis, ribosome as a common pathway for increasing total root length and root dry mass. So though we found a lot of DAPs, like 113 DAPs for NIL pair 6, we found only 3 DAPs, which is cytochrome B561 and domain containing protein, asparagine synthase, and thyrodoxine, which is highly abundant into the genomic region of the targeted genomic region of the NIL pair 6, while only 3 differentially abundant proteins again were found in NIL pair 9's targeted genomic region, which are 60 ribosomal protein L34, signal recognition particle 19 kilodalton, which is in short SRP19, and 3,4-hydroxy 2-butanone 4-phosphate synthase, in short DBPS. So we can see that though we found a lot of protein, differentially abundant proteins, only few are locating inside the genomic region, and I will explain it in my further slides how we can improve that. So further, we were interested to see what is the mRNA expression of these proteins, which are located particularly on the genomic region of the NIL pairs, because they are like very much conserved. So we found only asparagine synthase from the NIL pair 6 shows similar expression, means high abundance in both transcription and translation. On the other hand, for the NIL pair 9, SRP19 and DBSP showed similar expression, means lower abundance in both transcription and translation. So considering these both similar expression pattern in transcription and translation, we considered that these proteins can be the candidate protein biomarker for total root length and root dry mass in wheat. Further, we investigated the molecular pathways of the candidate proteins, and we have seen that the asparagine synthase are associated with the aspartate and glutamate metabolism pathway. So I have just zoomed it here. Here is the total pathway, and this is the specific pathway, if you can see that. So they are associated with glutamine also, L-glutamine, acetyl-glutamine. So they participate into different functions, but yet there is no clear explanation that they are contributing to the root trait improvement or like increasing root rate or decreasing root rate, which is the beauty of this protein. Again, the SRP19, which is the DBHP, that is associated with the riboflavin metabolism, and here is the complete metabolism of the riboflavin, and here of the protein, and here is the protein, and it is associated with the riboflavin metabolism. And the SRP19, you can see here that this is the SRP, and SRP19 is contributing to the protein export, which is very important for any growth and development of the organ. So here is the SRP19. So from the literature, it has been found that the candidate proteins which we have identified contributed to many functions of the root growth and development. For example, asparagine synthase maintains carbon and nitrogen metabolism in crops, and it also contributes to the ROS scavenging, which is important for abiotic stress tolerance, particularly in wheat. They contribute to the wheat root. And then again, DHBP, which is indirectly contributes to the root growth in Arabidopsis and maize. The signal recognition particle, or SRP19, which supports SRP54, which actually contributed to the drought tolerance in wheat. It has been found that due to the downregulation of SRP54, that drought susceptibility increase, while SRP19 contributes to the SRP15. So from the literature, it has been found that no clear information about these three proteins for controlling root rates in wheat. And here it can be the novel protein to explore further to improve targeted root rates in wheat. As I already mentioned, that though we found a number of DAPs in the different NIL pairs, but we did not get more DAPs into the target tiers. So what could be the reason? So as for example, in our case, we have developed the NILs up to F8 generation. So if we look at here, in F8 generation, the heterozygous background is only 0.78%, and we are suspecting that maybe this 0.78% heterozygosity contributed to the larger DAPs outside the genomic region. And we are proposing that if we increase the generation means advancement of for developing the genotype, if we advance it up to F16, we can see the heterozygous background is actually reducing 0.00%, and homozygosity increase in 99.7%. And therefore, we can expect more genes or more proteins inside the genomic region of the near-isogenic lines, because theoretically near-isogenic lines should have more proteins, or like more differentially abundant proteins inside their targeted genomic region. Therefore, we are proposing to increase the generation of developing near-isogenic lines to get more differentially abundant, or like more abundant proteins or genes inside the genomic region, so that it can help us to narrow down or select the reliable proteins, or more, giving them more avenue to choose which one we can go for. So in a summary, from these experiments, we have identified a number of differentially abundant proteins which are important for increasing total root length and root dry mass. And we also found 3 novel candidate protein biomarkers, like the asparagine synthesis. High abundance of asparagine synthesis can contribute to the increased total root length and dry mass. The lower abundance of signal recognition particle 19 kilodalton protein, and as well as 3,4-dihydroxy 2-butanone 4-phosphate synthesis can contribute to the increasing the total root length and root dry mass. Also, the molecular pathways that we have identified: cell redox homeostasis, structural molecule activity, protein binding, which can be important for growth and development, as well as the other metabolic pathways which are associated with these candidate protein biomarkers, for example, for asparagine synthesis, aspartate and glutamate metabolism, for 3,4-hydroxy-2-butanone 4-phosphate synthesis, riboflavin metabolism, and for signal recognition particle, protein export, and carbohydrate metabolisms are important molecular pathways for increasing the targeted root rates in wheat. So our paper has been already published in Journal of Proteomics. We have discussed our outcomes, and these protein data has been already deposited into the PRIDE. So if anyone is interested, they can look at the data of total protein data to use it. So in a nutshell, these three novel protein biomarkers and molecular pathways can be used in future with breeding. However, it needs the functional validation, and which I'm looking for. If I get the opportunity to do the functional validation in my next career pathway, I would like to do that. And then also, if we can investigate the protein-protein interactions, and also more investigation on the molecular pathways, how these molecular pathways are related with the root rate increase, and how it contribute to improve the targeted root rates. It can be useful in wheat root breeding, which is very important to increase the grain yield as well as climate resilience. Thank you all.
Okay, do people hear me? Thank you so much, Tanushree, for a great talk, very interesting. And thank you for the attendees. We have time for questions. And I will try to. Okay, so we have our first question. Thank you, Dr. Tanushree, for the nice presentation and nice work. I am wondering if you related below-ground biomass with above-ground biomass. If no, how does below higher amount of ground biomass contribute to yield?
Well, thank you for the nice question. Uh, in this particular experiment, I did not count the above ground biomass, but in the literature, it has been found that total root length is positively correlated with the shoot height. So that is one dimension that it can improve the plant height, and by uptaking more water and nutrients from the soil, it can contribute to the grain yield. Because if we think about, like, in a drought condition, if the soil water level is down, if the total root length is higher, it can uptake more water and nutrients, and thus it can contribute to the grain filling stages, and we can get a more grain yield.
Okay. The second question is, what is your challenge during your experiment by using hydroponic system in order to face wheat drought stress?
Well, I did not do any drought experiment, but the challenges of semi-hydroponic system was like to continue the circulation of the water and nutrient supply, because if you look at the system, I go back to that slide. So continuous supply of water and nutrients are continuing through the pump. So you need to actually take care of that. But I think that if you want to do some experiment on drought in semi-hydroponic system, um, you may need to use some of the chemical like bank, or like you need to stop it for a while. You need to turn off the pump for a while, and then you can continue the drought experiment in semi-hydroponic system.
Okay. Thank you for that. The next question is, in the root space economics, what could be the right proportion of total root length for optimized increased grain yield? So what is what proportion, I guess, of the, how does root length, uh, how can that be optimized for grain yield?
Well, this is a very tricky question. So total root length and grainal relationship is also dependent on the soil condition and also the climatic condition. For example, if the soil condition is like it has more water and nutrients in the surface soil, then the total root length ratio and the grainal ratio, which if I don't make any wrong assumption, it will be different. But when it is sandy soil or like the soil where the water and nutrients means water level is far below, then it will be different. I think I can better explain it later. Can we discuss it later?
Oh, sure. So there would be opportunity if you want to write an answer as well to follow up. Okay, so the next question is, thank you. Thank you for your answer. Did you identify any important SNPs for your candidate proteins? And how can a breeder use your biomarker in molecular screening of germplasm?
Uh, First of all, I did not use any SNP. So I tested the mRNA expression using SSR marker, means the primer was like SSR. That can be a future study. And yes, that's a good question, that how can anyone use this one in molecular investigation. So as we have already developed some primers which is associated with these proteins, and also we have the uh, sequence of the protein as well as the sequence of the genes, so they can use it for further molecular investigation. However, for use it as a practical biomarker for improving the root length and dry mass, as I have already mentioned, that we need to go for the functional validation. Otherwise, we can use that primer or like gene information from the paper.
Okay, uh, we have a couple more questions. Once again, the person who's asking the question thanks you for a great presentation and wants to know why you used the semi-hydroponics method instead of a fully hydroponic system, and and I guess how you established the system.
Okay, thank you for the question. So the semi-hydroponic system actually, as it allows us to accommodate more plants into a small space, first of all. Secondly, the system has been already validated that the root growths of this system, using this system, and the root growth in the soil is uh, the has a positive correlation. So therefore, like having this opportunity that already it is economic and already validated. That's why I went for that semi-hydroponic system rather than going for some full hydroponic system.
Okay. Um, can you talk more about the mode of data analysis that you used? What do you recommend for your study for future research direction?
For future research direction, I think that we can use these candidate proteins for CRISPR-Cas, or like we can do some of the functional validation using. We can do the Western blotting also, and like knockout of these proteins, and can see that if it is really working or not, so that the future study can be like genome editing, or further functional evaluation approaches.
Ah, okay, here's another person who's complimented you. Thank you, Tanushree. I love your presentation.
Thank you.
So my question is, if you could do something different in these experiments, what would you change?
I think I would not change anything. Rather, I could add something. If I had time, I would like to add some of the functional validation studies so that I can do something for the with breeders so that it can be used practically. Thank you.
Okay, that's the last question. I just have one quick question. Um, I wonder if you can elaborate more on the protein assay. So you do a protein digestion with trypsin, and then are you using, does that go onto a column and then mass spectroscopy, or how are you actually identifying the protein sequences?
So after protein extraction, I'm just going to that slide. So, uh, if I don't misunderstand you, Joshi, you asked me that how did I get the proteins, right?
Yeah.
Okay, so after
So,
protein extract, yeah,
yeah.
no, please tell.
Oh, yeah. So I see the protein digestion, and then
Mhm.
can you describe more about, I guess it's called max quant and label-free quantitation?
Yeah, so after protein digestion, actually we used that mass spectrophotometry definitely, and we used orbit, and from that peptide data of that information, we identified that peptide sequences using that MaxQuant software. And further, actually, using that peptide data, we used Perseus software to identify the differentially abundant proteins uh, using student t-test, which showed significant difference between the proteins, which was in between the isolines. So
I see.
after
Okay.
protein digestion, definitely we went for the mass spectrophotometry.
Okay. I think that's where what I missed in that slide.
Yeah.
Okay. Um, and then it looks like there's a follow-up question about the hydroponic system, and the audience is very interested to know more about how you established this semi-hydroponic system.
So the semi-hydroponic system has been identified one of my supervisor in Long Chen. So it is like, the trash bin. So they used that trash bin, and the system has been developed as like you can see that this is a glass bar. So this can be used here, and then the nutrient composition is different from plant to plant. So that has been also like optimized by me and my supervisor for the wheat grown for the wheat plant growing. So if you want to establish this semi-hydroponic system, I think I can recommend a paper after this webinar. I can share you the paper where the detailed establishment of this semi-hydroponic system has been discussed by Inglong Chen. That will be helpful.
Oh, okay, that would be wonderful. Thank you so much for that. Okay, that's we don't have any more questions. So once again, I really want to thank Tanushree for a very interesting and excellent talk, and I want to thank everybody who attended to take their time and join us today. Um, I will remind you again, so there will be another webinar next month by Simon Griffith, so please uh, sign up for that. And once again, thank you all, and thank you to Nushree, and have a great day. So I appreciate your attendance. Thank you so much.